Good morning, good morning, good morning. And there you go. With a music theme, pre-class music theme that was throughout the term based on diversity, based on Canadian artists, and for a long time wasn't going to include the Beebs. Smith responds to feedback, hello Jack, and here we are. The Beebs, proud, but of course being from Stratford, he gets his peaches from Niagara, not from Georgia, whatever the song says. So today's going to be a little bit different. I can't believe, as I look out the window that's open at this time of year, that we have survived and gotten here together, arrived here together. So let's celebrate that. And we're going to do that in a couple of different ways. One of them is we're going to review with a somewhat discursive part where I'm going to kind of go through what I feel to be a lot of the highlights of the course and the themes of the course. Things that you want to consider as you're studying for the final exam. We'll talk a little bit about the final exam. We're going to do it a little bit actively then because we're going to use a bingo game. So if I say a word in that review of the course and you have the word on your card, you line 5 of them, bingo. That's how bingo works. We all remember that. You remember being in crowded bingo halls? In my case, smoky crowded bingo halls. So first of all, with the word, the letter fine, 2, 4, 6, 8, 10, 12. So you've got them all. Does anyone have an idea of what the word is? Put it in the chat, what the phrase is. I'll pause. I'm going to clear your beautiful thoughts. And we'll expose. So those are the letters. And that's the phrase. This is the theme. You've done a whole bunch. You've taken this course and all of your other courses in an absolutely unique and crazy time. And you've got to this point. You've got this. And that's going to be the theme for today, the games and the activities for today. And so here we go. So first of all, I'll tell you about the bingo. There's a welcome message on CourseLink, and that links to a PDF. The PDF has 5 different arrays like this one. This is Team Tardigrade. There's only one PDF. There's 5 different bingo cards within it. And what you can do, if you're willing to play along, is download that card. And you're going to put yourself in a team based on your surname. So I'm Smith, right? And so I would be Team Spiny Echinodermata. If your surname is Nelson, you are the Ornithopterans. If you are the Eames, you're the Nematodes. If you're the Aarons, you're the Tardigrades. And if you're the Zoo, you are the Molluscas. And marks off for Smith for spelling mollusca wrong at this point in the course. What the heck's going on with that? So, does that make sense? Now these teams, you're going to be in them for both for Jeopardy and bingo. So did those rules make sense for the review? If I say these words, like for example, if I was to say cephalocardata are deep within nature, but they're kind of like ketognaths, but I love the urochordates, then boom, Team Tardigrade wins. Clear as mud? Ooh, we begin. Anytime throughout the lecture, you got it. So, here we are. This is us. This is you kind of swinging and climbing and taking your drone now, your high-powered drone, and flying it through the trees of life. And we've done even better than this. Really, the only one of these branches that we haven't spent any time at is our own, because who needs it? It's a special interest group. You took a whole course. I don't know why we have a whole course dedicated towards the vertebrates, but there we go. So within all of the other nearly 3 dozen tips of this tree of life, fewer than 3 dozen, but nearly, we're going to talk about them together in that last lecture exam. Monday, April 26th, it's on CourseLink. The questions are going to be, so far it's just like all of the other quizzes that you've had so far. You're going to see true-false questions as before. They're going to see multiple choice questions this time, and short answer questions where you're going to, so that's a little bit different. The exam is going to be open for much longer than what we would kind of budget for the 2 hours that we would normally budget if we were reserving a room on campus for you to write the exam. The exam is going to be opening up on Monday, April 26th at 8 a.m. It will close at 10 p.m. These are Eastern Daylight Times, Guelph times, the times where we are right now. Well, my bias, I apologize, where I am right now. You have 4 hours once that is started, so everyone has double the amount of time that I imagined in writing this exam. So, remember the theme. You've got this. When will Marks be back for the second lecture quiz? Very shortly, like today. I'll open those up today. You did very, very well. So here begins kind of the quick review. If you've got your bingo cards, you got a dabber, that would impress me. If you've got a dabber, please tweet it or send me an email. But I'm not expecting dabbers. So basically, one of the biggest hints for this course is as you surveil and fly your drone through those trees, those branches of the tree of life, remember to try and contextualize phylogenetically the functions of all of the taxa that we've dealt with today. Know the relationships between the groups and the characteristics that we use to define the groups. That can be like a guiding principle as you're going through the studying for this. Okay? Now, some of the themes. So starting here on the top left-hand side, there's been a bunch of different narratives that we've covered throughout the course, like back into the first week of January. We've seen different taxa struggle with how to be a parasite, how to successfully be a parasite, and how to see, how to respond to visible and non-visible lengths of radiation. Adaptations according to symmetry or according to feeding style and according to and symmetry. We've seen different adaptations associated with cephalization, and being able to talk about those and use examples from different taxa that we've seen, those would be a really strong kind of a studying strategy. Phylogenies, thinking about things in a phylogenetic context. So focus on, remember some, kind of dive into some of the controversies. Whenever I said, when I was taught this course, we thought this, and now we thought this, and maybe there's even an ongoing controversy. So what are the morphological and molecular data on either side of that controversy? What are the consequences towards one of the hypotheses and the competing hypotheses as pertain to some of the evolution of some of those traits? You remember back in the first lecture quiz, we talked about ctenophores and sponges and what are the consequences towards the evolution of the nervous system that are dependent upon where we imagine and where we model ctenophores sitting. What is the controversy and what are map those major topics? Think about being able to mapping topics onto that phylogeny. Little, again, bingo dabbers. Functional topics in the bottom left-hand corner here. So musculature and body walls. Think about how things move, how they respond to stimulus in the environment, how they molt in some of the, all of that diverse ecdysozoan clade. How do the tardigrades do it? How do the onychophorans do it? How do they move? So different strategies for locomotion. Again, we could think about the hexapods. Look at all of those different strategies for mobility within that group. Excretion, with the Malpighian tubules, where do they sit? How many times do we see the Malpighian tubules show up? Think and remember, in this last third of the course, we're talking also about the mollusks. This is where the mollusks and the arthropods and all of those diverse groups occurred this year. So think about different adaptations of the shell, reductions and specializations of the shell, specializations of the mantle, of the foot. Evolution of appendages, serial homology, uniramus versus biramus appendages, specializations across the across the anterior-posterior axis in terms of tagmatization. These are all themes that you should be able to kind of map onto the taxa that we've seen. So closed and open hemosystems, trachea versus gills, the partitioning of the coelom, cephalization, nervous system evolution, we talked about cephalization, pentaradial symmetry, flight in the arthropods, eyes in the arthropods, patterns of cleavage, what it's used for, whether it's useful, whether it's not. Where have we seen asexual versus sexual reproduction? Where have we seen metamorphosis? What's in line or what's tied into metamorphosis? Thinking about apomorphies, changes in those synapomorphies that define major lineages. So when we think about the radula, how plastic is that? The mantle, how plastic is that? The foot in the mollusks, how plastic is that? The answer is very plastic in all cases. Arthropod mouth parts. One of the big themes that we've kind of come back to again and again is to introduce you to a group and to say, here is a synapse. Here are the kind of the characteristics that we use to define the group that are unique to this group, and here is where they are lost in groups in taxa that are contained within that larger phylum, for example. So loss and gain of traits. So where have we been, and how long have we been there? So this is back to the 21st lecture. So the specialization of this third, and I see there's a question in the chat there about the final lecture exam. It's going to focus on the last third of the course. If there are thematic questions like parasitism that you want, for example, that you want to bring in examples from earlier, that's fantastic. But the specific questions in the true, false, or the multiple choice, those will involve the last third of the course. So I think from, if I remember correctly, lecture 21 onwards. So basically mollusks until now, until today. So we talked about the testaria. Here's an example of mapping a term onto a phylogeny. Is testaria monophyletic? It's not. We talked about the generalized mollusk. We talked about the plasticity of features like the foot. A reminder, I see that in the chat. How do you use the bingo card? If I say a word, if some of you out there have the phrase mantle on your bingo card, talk it off, cover it up, I've just said it. If some of you have testaria, cover it up, I've just said it. And the teams, which card do you choose? That's based on your surname. So the very first, there's A to O, your tardigrade. E to I, your nematodes. J to N, your onychophorans. O to S, your echinoderms. T to Z, your mollusks. And I'll post that again before we get to the Jeopardy game. All right, so within the mollusks, again, in mollusk, oh my God, mollusk. We could have spent the whole class on mollusks. We could have spent half the year on gastropods alone. We talked about aplacophorans. With a really reduced foot. So again, reduction of one of those defining traits. And the polyplacophorans, which have eyes in their shell. What are you talking about? Polyplacophorans, tegumentum. We've talked about gastropods and torsion. And the success that permits in terms of moving and manipulating a big, cumbersome, but protective home. The monoplacophorans. And we listened to actual scientists, actual nerds, live, captured live on camera, discovering monoplacophorans seen alive for the first time ever in the class. I love that. That excites the hell out of me. We talked about bivalves with their lost radula in this case, and their byssal threads connecting to the substrate. We talked about cephalopods, and probably for many of you, we didn't spend enough time talking about cephalopods because cephalopods are kind of amazing. And so how did we not spend more time talking about siphons and their shifted orientation in terms of ventral, anterior, their arms and their tentacles, their eyes that are just incredible to us. And we talked about scaphopods, tusk shells. We then jumped into the ecdysozoans. We talked about the ecdysozoans and the panarthropods and the cycloneuralia. But principally this group on the top, we didn't use it a lot. We jumped into it right at the very end when we talked about the minor phyla. Remember, when we're talking about those minor phyla, what were some of the themes that linked them together? They were small. They tended to be marine. They tended to be discovered only in the last 50 years. They tended to be commensal or parasitic. With those panarthropods, though, oh my God, the panarthropods. There's just so much to have talked about within the arthropods in general, but the nematodes, the diversity of nematodes, circumoral rings. We talked about nematophorans, the things named Nema, the Nemertean worms within the spiralians, and then nearly featureless Gordian knots, or the nematophorans, emerging out of their insect host as they bring them, mind control them to come towards water. Mind control by parasites, that's definitely a theme. Tardigrades, we probably could have spent more time talking about tardigrades. For sure we could have, because they're perhaps the cutest thing. But then I hear you say, Team onychophoran, that's definitely the cutest thing. The lopopods within them, the hydrostat that allows them to kind of wander along. Their claws, the stacked constituent elements that comprise those claws that link them to hallucigenia. Oh my God, I want the whole class about that. But no, I don't. I want to talk about arthropods, and I want to talk about the success of the tagmatization of that body plan and the specialization of the legs. That basically, what are arthropods? They are a bunch of jointed appendages. We looked at specialization of the crustacea, this group, this non-monophyletic group that often have biramus legs, not just uniramus legs. We then jumped into the millipedes and the centipedes, and we talked about the Thomas-Sherry, where the organs on the head are probably hygroreceptive. We spent not enough time there, but then we moved on to the chelicerates, and we talked about the chelicerates, even the intimate ones that live on you. We talked about pedipalps and walking legs and tagmatization down to basically a head and a cephalothorax. Spiders, all the intimate, gorgeous ways that spiders can have devised to capture their prey. We then spent some time talking about hexapods, not nearly enough, but oh my God. Hexapods, social things. So Malpighian tubules again, Malpighian tubules and convergently evolved traits. That's kind of another theme, things that we've seen throughout the course that are not due to shared ancestry, traits not because of a shared common ancestor, but traits because perhaps of a shared strict environmental stress, and in this case, perhaps moving from a marine ancestral environment to a terrestrial environment. The apparently independent evolution of Malpighian tubules in the hexapods, in the some lineages of chelicerates, in the, well, we talked about it, in the millipedes, and in something else that I'm slipping my memory right now. Then we jumped to our own group, which doesn't interest me a lot. That was my bias that I introduced, but then I hope I got us, I shared some of your excitement about the echinoderms, about these apparently bilaterally symmetrical things that have pentamerous or pentaradial symmetry as adults, this incredible adaptation of the water vascular system. We talked about crinoids and sea stars, sea cucumbers and sea urchins. Then we jumped into the hemichordates, the cephalochordates and the urochordates, and we talked about, within the urochordates, the only colonial chordate. That's kind of amazing. And then I sang to you about the cephalochordates and the amphioxus, and I promised the internet that I wouldn't do that again today. So, as you're reviewing for this, my suggestion for you, oh, bingo, which team? Bingo. Bingo. I see a bingo called. That's amazing. Team Tardigrade, hooray! And the prize is joy, inner calm, and peace. So, one of my suggestions as you're studying for this class is really not to just read things over. Do that. Start there. Read your notes. Read the parts of the text that pertain to those notes. Distill those. Write them out again. Get them down to smaller and smaller bits. But that's not the end of your studying. That's the beginning of the end. Make your studying past that point active. And by active, I mean study together. Try and write questions for one another. And that's where I'm going to try and model with this Jeopardy game, which I'm going to give you a link to later on so you can see the game that I wrote, and then you can write games yourself for your friends. There's some other ways that you can make yourself active and kind of integrate or associate with course material. One of them is the fact, and these will all be put in the chat on CourseLink later on, but there is, some of you found it already, but there's a playlist of all of the lectures if you want to go back and spend 36 hours together in this room with the portal to Narnia behind me. We can do that. You can also, any of the videos that I've used in the class that are on YouTube, I've arrayed on this playlist. I think there's now about this is 326 or 350 videos. So that's like, I think 36 to 48 hours of film that you could watch if you're interested in that. And then also if you're interested, if you use music to study and you like some of the tracks in that Canadian and diverse and equitable attempt to be equitable in terms of reflecting Canadian music, there's the 80 odd songs that are in the playlist from the start. And now we're going to start this game. So here's how we do it. We're going to, over the remaining time in class today, I'm going to be Alex Trebek, and we pause for a moment to remember the Canadian Alex Trebek, and we are going to play Jeopardy, a Jeopardy-style game, okay? Okay. So, those surnames. So, the bingo game, don't lose that team. The team Tardigrade, for instance, you're still all united in your disparate spots around the planet. I'll have a random draw. I'll think of a number. The team that answers in the chat that gets that number first, they will they'll be able to answer the first question. I'll start it. I'm still making these up in my head as we go, but we'll start with warm-up round for 100. I'm going to reveal the clue, read it aloud, after which, and this is where it departs from Jeopardy, any team member from any team can use the Zoom chat, and this is going to be different. So this isn't the course link chat. I've left Zoom chat open, so I'm going to try and keep these multiple windows, and I'm trying to keep your number of windows you have open minimized. So, but phrase it as a question. So, for example, if a contestant were to select new things about cnidarians, the resulting clue would be the protozoan member of a symbiosis, one thought to only have two partners, to which the correct response would be, what is a dinoflagellate? Okay, so phrase it as a question. And if you can, if your Zoom name has your surname in it, I should be able to figure out which team you're on, but you can throw that in as well. So some of that, like, you could write out what is the, and copy it so that you can just paste that into the chat real fast to increase the speed with which you can answer. It's just first in, first served. So whoever gets the correct, I'll add that clue's value to the team score, and then they can select that team in the chat can select a new clue from the board. And so really this last bit, I'm just realizing there is going to be no incorrect responses because it's just going to be open and first come, first served. Okay? I think this makes sense. No penalties for having wrong answers at all, so you're not going to lose anything. Questions? So let me first of all say hello in the Zoom chat. Here we go. And let me pull this up and ask you, did I just change your screen so that it is one of now, you can see a URL bar at the top? I think I should have. Okay, and I see you answering. Good. Now I think these answers are going to come in super fast. So remember, what we're doing here is kind of modeling how I would suggest studying, okay? So I'm thinking of a number. Is everybody ready? I'm thinking of a number between 1 and 10. First team in the chat. Okay, hold on. Oh my God, this is going to be crazy. Ocampo, ABCDLMNO. Echinoderms, you got it. Team Echinoderm, the number was 7. So here we go. Now, we've got in the warm-up round. First clue. No single member of this group has 1,000 legs. No, oh my God. Look at this. Okay, Juliana Magdish. So team. Team Onychophorans. Correct. Hooray. Well done. So what is the millipede? Yes. Okay, so whoever, Team Onychophorans, which one would you like? Tell me where to go. Somebody from any representative from Team Onychophorum. Tell me where to go. Pop culture for 100. Perfect. That's what I saw first. All right. The Star Trek's USS Discovery was threatened by this invertebrate. Tardigrade is correct. Emily, Team Mollusca. Hooray! There we go. That's correct. The tardigrades. And they kind of use some incorrect interpretation of horizontal gene transfer to make a villain. This is going well. Okay, Team Tardigrade. No, what am I saying? Team Mollusk, please tell me where to go next. Pop culture 400. There's some strategy going on here. A fossil of this invertebrate group was used to inspire the facehugger morph from the Alien movie. I see answers. Crustacean, no. Arachnid, no. Crustacean, no. Echinoderm, Tullus, Emily, Mollusca again. Well done. Here we go. That's right. It was a crinoid. It was a crinoid that was sitting on top of the flipped over upside down. Now, let's escape. And somebody from Team Mollusk, tell me again where you would like to go next. Pop Culture 5, I see. What insect lifestyle inspired the chest-bursting scene from the movie Aliens? Insect lifestyle. I see parasite twice. Parasitism, it's more specific than that. Parasitoid, Shaw, Courtney Shaw, Team Echinoderm. Well done. There we go, you're in the game. The chest-bursting scene, of course, really is quite modeled upon that, that parasitoid lifestyle. Okay, here we go. Now, somebody from, what, I just gave the points, yes? Now, somebody tell me in the chat where to go next. Somebody from the team that just won. Pop Culture 200. This mollusk is the most maritime of the sigils of all of the Game of Thrones houses. Maxwell, Hannah, super, the squid. Well done. Maxwell, LMN, Echinoderm. No, LMN, Echinoderm. There we go. The Kraken is, of course, a giant squid. Here we go. Somebody from the Onychophorans, give me a place to go. Mollusks 200. I like it. This part of the molluscan body secretes the shell. Not even time to start it. The mantle. Let me scroll up to the first one. Oh my God, that's all of you. Robertson is the first I see. RS Echinoderm. Awesome. The mantle or pallium. Or pallium. All right. Give me a place to go. Mollusk 400. This is the morphological hallmark of the trochophore larva, a larval stage characteristics of most mollusks. Free living, villager. No, free living. Kitty. Oh, I love it. You're thinking of like all these. Okay, and the answer there was the prototroch, the mustache. So the question, whoever got the last one, I don't think I saw that. Tell me if I'm wrong. But tell me where to go next. Team, I just won. Pop Culture 300. Tom Hanks and Ron Howard have made a horrible movie about this sequence of numbers that you may know is common in the creation of molluscan shells. Courtney Shaw, Fibonacci, that is correct. There we go. The Fibonacci sequences. So echinoderms. All right, tardigrades and nematodes, we got to see you. Let's get in there. Okay, so echinoderms, tell me where to go next. Pop Culture 600. We're using up the pop culture. I like it. The horror movie The Bay was about tongue replacement zombies and was inspired by this vertebrate. Oh, Alex too. Well done. Team Mollusca. It is an isopod. Hooray! An isopod crustacean. Well done. Team Mollusk, give me a place to go. I see lots of correct answers there for isopod. Well done. Actually, echinoderm, I think I just might have messed you up to some points, but come on, Team Tardigrade. Should we give it somebody from Team Tardigrade? Or Team... Okay, spice her up, Panarthropod Panda 100. Here we go. Tardigrades from Antarctica were recently revived after more than 30 years. What is the name these samples were given, although they weren't awakened by a kiss? Sleeping Beauty. That is correct. LMNOP. Team Echinoderm again. Sleeping Beauty I see across a bunch of different groups. JKLMN. And I'm scanning. Okay. Sleeping Beauty. Pilon is Echinoderm. Well done. So, Echinoderm, tell me where to go next. Hexapod 200. I like it. These tiny hexapods are characterized by recessed jaws. Recessed jaws. Yes, Margaret, Collembola and springtails are correct, but what is the group? Sherry for Team H. Hooray, Entognates. I love it. A, B, C, D, E, F, G, H, I. All right, nematodes. I think you brought a ringer. Here we go. So Team Nematoda, tell me where to go next. I like it. Team Nematode. Well done. Somebody from Mollusk 100. Molluscan Madness. The structure of a molluscan gill is often compared to this flying adaptation. Ah! F, G, H, I, J, K. A, B, C, D, F, G, H, I, J, K. Onychophorans. That's the first one that I see. That is, in fact, correct. It is the feathers. All right. Onychophorans, come give me a new place to go. And those are all good words about it. Bipectinate, yep. So tell me a place to go. Where have we not been? Crushing chelicerates. We have deuterostomes. That's right. 200. I like it. A protrusible jaw apparatus in the echinoidea was named for this Greek philosopher. Yes, that was right. O'Donnell, echinoderm, Aristotle. Wah, wah. There we go. Okay. Give me some more. Give me the echinoderms. Somewhere to go. Hexapod 300. Hexapod. Tiny springtails are springing all over your lawn right now, and they're using this organ to do it. Nematodes. I see one of you who has, I see a bunch of this, and I'm going to then use my privilege as Alex Trebekian, my mustachian privilege. I'm going to do that to the nematodes. It is the furcula. Furcula. What the furcula? They are using the furcula. So, nematodes, you're on the board again. Tardigrades, come on, you can do this. You can endure, right? That's your namesake. So, Team Nematodes, tell me where to go. Chelicerates 100, crushing it. This giant arachnid was a major problem for Sam and Frodo in The Lord of the Rings. Yes, it is a spider, correct. And I'm looking for a name. Because Tolkien gave the spider a name. Yes, I see. All right, echinoderms. I see a bunch of the echinoderms in there. It is Shelob. All right, let's go and tell me where to go, echinoderms. Hexapod 500. Articulated appendages have been an evolutionary leg up for arthropods, but this appendage is not derived in the same segmented fashion. What appendage? F, I see Faulkner. A, B, C, D, F, nematodes. It is wings. That is correct. Hooray! The analogy I, of course, gave for wings was that they're like your muffin top, and like you could fly. It's a derivation, an outfolding of your body wall, and you're going to then flap around by pulling on muscles that are contained or attached to the the ventral and posterior, dorsal and ventral parts of your body. Incredibly unhelpful. So that was, I think, nematodes. Give me another. Nematodes, you're on the board. Actually, you're doing quite well now. Give me a place to go. Warm up 200. Oh, I like it. Good strategy. Phylogenetic grouping that helps us make predictions is called a... Yes. All right, Team Tardigrade. You're on the board. Woo! Monophyletic. I love it. I love it. I love it. I love it. Way. Hey, hey, hey. Now this begins the late game run. Tardigrades, use some gorgeous legs and take us into the game. Where are we going to go? Things NEMA 100. Nemerteans have this fluid-filled chamber that contains a proboscis. Not a coelom. Not the Malpighian tubules. All right, we're going to call that one, and the tardigrades retain the capacity to get the next clue. This was the rhynchocoel. I was pointing at my nose. This is the rhynchocoel. Okay, team tardigrades, tell us where to go. Team tardigrades, you're still out celebrating your victory. Warm up 300. I see that, and here we go. The geological period of the limestone that Guelph is built upon, once a tropical sea floor. I see Ordovician and Cambrian, no and no. Devonian, no, but you're in there. Ah, yes. Silurian, F, A, B, C, D, E, F, nematodes. It is the Silurian. All right, somebody from Team Nematode. Tell us where to go, please. Hexapod 100, hexapod hijinks. The world doesn't need to fear giant ants because of this physical action by which insects breathe. Yes, I see an answer. Trachea. Where did I see that first? Diffusion. Yes, that's what I've highlighted here. I see Carson. I'm going to award that to the nematodes. That's correct. It is diffusion. Large insects would, of course, die a slow death of lack of air. Hooray! All right, tardigrades. Come on, onychophrans. Come on, tardigrades. You like lopapod bearing walking. You're like, oh, some of my favorite groups. Let's go. Team tardigrades. Wait, what? Carson, A, B, C, D. Yes. Yep. Okay. Tell me where to go. Team tardigrade. Mollusks 300. This major group of mollusks does not possess a radula. Carson, whoa, you're on a roll. Yes. Bivalves. That is correct. That's the first correct one that I saw. Tell me where to go again. Come on, onychophrans. Start using that elastic instability. Warm up 400. The abandoned mill in downtown Guelph. That was host of a termite invasion. We're going for your Guelph knowledge here. It is a paper mill, or it was. Not King's Mill. Yes, Courtney Shaw, Goldie Mills. S, echinoderms. It is Goldie Mill Park, and you can go once we're not sheltering in place, and you can explore there, and it's kind of lovely. Some people in better times and different times have their wedding pictures taken there. Okay, Team Echinoderm, tell me where to go next for the next clue. Hexapod 400. The ground plan for the hexapod mouth has been modified dramatically in many species. The original model was designed for this. Echinoderms. Oh, hold on. I see tardigrade. Yep, herbivory. Herbivory. All right, echinoderms, you're in. Now, echinocophorans, please. Let's have a sit down here. Onychophrans, meet me over here on the side screen. You can do this, right? Like, you know what you're doing. All of these questions, just quicker on the paste in, write out on your typing on your keyboard. What is the? Select it, copy it, and then you can paste it in. Then you're fast. You got this. You can shoot. You've got it. All right. Tardigrades. I think you just got that one. Tell me where to go. Team Panarthropod 200. This is the process by which bacterial genes were thought to have moved into the tardigrade genome. Yes, Courtney Shaw. Horizontal gene transfer. Where are we? S. Echino. That is, of course, correct. And then 9 days later, we thought, no, hold on. In fact, that's an analysis of bacterial contamination. So, Team Echinoderm, tell us where to go. Horizontal gene transfer. Team Echinoderm, use those spiny dermises. Tell me, who do we got most left of? Things Nema and Deuterostome Delights. Warm up 500. Okay. The flashing of the disco clam serves this purpose. I see. Scaring off predator, anti-predatory. Yes, yes, but this is the answer I'm looking for is more specific. Yeah, warning is getting closer. Toxic warning, yes. Now, who was that? Toxic warning. O'Donnell. There we go. An honest warning of its toxicity. And that is O into the echinoderms. I think I should have looked at how many students were in each one of these categories, but you're doing very, very well. Because I have a suspicion that Team Tardigrade or Team Onychophoran, maybe both of you are out there with fewer members of your team. Okay, so echinoderms, tell us where to go. Which part of the board are we going to use? Chelicerates, I love it, 300. This taxon may just have exploded our understanding of a single transition to terrestrial life within the chelicerates and of the understanding of book gills to book lungs. Hold on. I see Faulkner. I see, and I'm cherry picking right now. A B C D F G H I J K L K. Ugh. I'm cherry picking. I'm definitely cherry picking. Good answers, everybody. Okay, I was cherry picking. Now I'm going back to the very first one. Alex, molluscan, you got it. It is the horseshoe crabs, the xiphosurans. And this is, of course, still being contested by different groups that are examining the same evidence. So, Team Mollusk, give us your best hypothesis of where to go. And Team Monty Coffin, come on, squirt that out of your lopopods. Stick-to-itiveness. Pop culture, 700. The Simpsons crazy cat lady may have actually been a person infected with this protozoan. All right. Onychophorans, kraut. I see that. Toxoplasmosis. Hooray. And remember, this is what I was saying in terms of there's an emphasis on the last third of the course in the exam that we're preparing for. But if you want to bring in adaptations as you talk about themes like how to be a parasite, you can use examples from throughout the course. And so this would be one of those cases. We talked a lot about different parasitic adaptations and the manipulation of their host in many taxa, including that protozoan one. Okay, onychophorans, tell me, you're back in, look at this. We've got a pretty even board now. Where are we going, Team Onychophoran? Mollusk, 500. This molluscan class was thought extinct until recently and is characterized by the serial repetition of organs. Alex, that was very fast. Monoplacophoran is correct. T is apropos, I suppose. Team Mollusk, that is correct. And this was, in fact, how they demonstrated when they were flying that ROV over that strange shell that they found very, very, very deep off the coast, I think of the Pacific coast of Costa Rica, I think. And they saw the paired, serially repeated gonads and gills, which is unique amongst all of the molluscan groups. Okay, Team Mollusk, tell us where to go. Team Mollusk is still celebrating. Tell us where to go. What is the next clue? Mollusk 800. Okay. Yes, there's a strategy now. The Great Lakes Brewery IPA label features a cephalopod. Octopus wants to fight IPA. I think it's delicious. Which of the principal molluscan synapomorphies is most reduced in this class? JKL. Onychophorans, I see it. Leora Samira Melamed, what is... though I saw it earlier. I saw it from someone from Team Onychophorans. I saw the shell. Onychophorans, you're in it. You're in it to win it. Okay, Team Onychophora. Tell us where to go next, please. Chelicerates 700, crushing it. The blood of what group of chelicerates is behind a multi-million-dollar health industry to test all medical equipment for contamination? JKLMNOP. Echinoderms. Pilon, yes. It is the horseshoe crab, and I see lots of you got that. That's amazing. Well done. All right, echinoderms. Oh, I see that. You remember the strangest things. I don't know if you remember the strangest things. It's just when you're talking about this group, you get to learn about the strangest things. Last couple of minutes here. Let's call this maybe the last question so we can sum up. No longer the strongest natural material. Spider silk is this kind of compound. What is spider silk? That is right, Alex. ST, mollusk. It is a protein. Well done, everybody. Awesome. Okay, so we have a pretty close game there. Well done, all of you. So Team Echinoderms, you pulled out the win at the end. Put your right hand up, pat yourself on the back of the head. Team Mollusks, that was a great big charge at the end. Onychophorans, nematodes, and tardigrades, you made a good play of it. This is a fairly evenly distributed board. Well done, all of you. And so what I will do is, as we begin the finish here of this particular shared time together, I'll tell you two things. One of them is I'm going to post, there's a PDF of this exact board that I'm going to put up on CourseLink in a second. And the other is to encourage you to think of yourself as discoverers of this, because we're still at such an exciting time in the history of life where we need you to be excited about the diverse things that we share the planet with, because we're destroying them at a crazy rate. And we now finally have tools to do that. And as you're doing it, think about the places that you've come, and also continue to pat yourself on the back. This has been an honor and a privilege for me and for Sherry to spend all of this time with you. So thank you in this kind of unique and crazy year. You've done really well in a crazy, crazy time. So as you're studying, be kind to yourself and be kind to others, and remember that you've got this. That's the theme for the course. So I'm going to stop the recording. I'm going to turn off the video here in a second, but if there's anyone who's got some outstanding questions that I haven't managed to answer, I'll answer them, and I'll do that back on Zoom here, but I'll also do it in the chat that's on your course link. So, well done you. Well done you. And thank you for all of those things in the chat. That's lovely. Yay. Okay, I'm going to pause video and audio. Oui, monsieur.
Are you done working?
Non, pas maintenant. Mais je ne suis pas, les étudiants n'est pas ici maintenant. As-tu une question?
But I'll need your help in one short sack.
Okay. Avec quoi?
Building.
Okay.
Something.
Okay. So, peut-être j'ai des questions ici de mes étudiants, so peut-être 2 ou 3 minutes. Okay?
Okay.
Okay.